Hello and welcome back to A Level Biology Help. Today I'm going to take you through the nerve impulses section for AQA A Level Biology. Also, near the end of the video I'll be going through a few exam style questions and I'll be explaining their mark schemes. Also, as always, there will be timestamps in the comments section so that you can skip to the different sections of the video if you do not wish to watch the whole thing. Right, so let's get started. So this is the structure of the video today. So first we're going to talk about the structure of a myelinated neuron, then we are going to talk about the resting potential, then action potentials, the refractory period, and then we are going to talk about the factors affecting the speed of conductance or the speed of impulses. Right, so let's make a start with the structure of a myelinated neuron. So this is a myelinated neuron. You might have already seen images like this. So this is a neuron or a nerve cell. So we've got the different parts. Here we have the cell body. I'll just get my pen tool out. So this main part here is the cell body. This contains the organelles that a normal eukaryotic cell has, so the nucleus, mitochondria, etc. The job of the cell body is to produce proteins and neurotransmitters. Neurotransmitters will become more important when we talk about synapse transmission in another video. Next we have these kind of extensions that extend from the cell body. These are called dendrites. The job of dendrites is to carry action potentials to surrounding cells. Now don't worry if you don't know what an action potential is. I'm going to talk about this later on in the video. Next we have the axon, which is the long conductive fibre that runs along the neuron. The job of this is to carry nervous impulse along the neuron. So it has a long conductive fibre. Next we have the myelin sheath. Now the myelin sheath is made up of Schwann cells. So each of these individual kind of cells here are called Schwann cells, and the whole thing makes up the myelin sheath, which is kind of like a protective layer, kind of like. Now the myelin sheath is a lipid, so it does not allow charged ions or the impulse to pass through, so it kind of acts like a insulation. And the gaps between each of the Schwann cells are called the nodes of Ranvier. Right, so now we're going to talk about how nerve impulses are conducted. So I'm going to start with resting potential. Resting potential is the difference in electrical charge between the outside and inside of the neuron when an impulse isn't being conducted, so when there isn't a stimulus to send impulses. Resting potential is about minus 70 millivolts because there are more sodium ions and potassium ions outside the neuron. So here we have a simplified image made by myself. So here we have a neuron here, the inside and the outside. So here we have 3 sodium ions outside and 2 potassium ions outside as well. This means that the resting potential is negative at minus 70 millivolts because these are positively charged ions, and the positively charged ions are outside the neuron, so this means that the inside of the neuron is negative. But how is resting potential maintained? The resting potential is maintained by what we call the sodium-potassium pump, which involves active transport of sodium and potassium ions, so therefore requiring ATP. So here we have an image of a cell membrane of a neuron with a sodium-potassium pump. So the outside and the inside of the neuron here. What happens is that 2 potassium ions are actually pumped into the neuron, and the 3 sodium ions are transported out of the neuron. Now you're probably wondering, well, if the potassium ions are transported into the neuron, then that gives the inside of the neuron a positive resting potential, not a negative one. Well, the thing is, Because the potassium ions have been transported into the neuron, this creates a concentration gradient of potassium ions inside the neuron, and also a concentration gradient of sodium ions outside of the neuron. This means that the potassium ions can then diffuse out of the neuron again, so creating a negative resting potential. And also the sodium-potassium pump becomes less permeable to sodium ions, so they can't be transported back into the neuron, so therefore maintaining the resting potential at minus 70 millivolts. So now I'm going to talk about action potentials, which is how a nervous impulse is conducted. So the first step is that when an impulse is received from receptors, the sodium ions bend and they stretch, so they open. This means that more sodium ions can enter the neuron as the membrane has become more permeable. This causes depolarization. The key term here is depolarization. Depolarization is basically when the inside of the neuron becomes more positive. So the charge becomes more positive as the sodium ions have a positive charge. If the depolarization reaches the threshold potential, so this will be about minus 50 millivolts as the sodium ions increase the charge inside the neuron. This means that voltage-gated sodium ion channels are activated, which leads to an even higher influx of sodium ions, so a rapid amount of sodium ions enter the neuron. This causes an action potential. The action potential happens at around plus 40 millivolts. Now it's important that you remember these approximate numbers. So when the depolarization reaches the threshold potential, even more voltage-gated sodium ion channels are activated, so there is a rapid influx of sodium ions, so this causes an action potential which occurs at plus 40 millivolts. So when, approximately plus 40 millivolts is reached, the voltage-gated sodium ion channels close. Whilst this happens, voltage-gated potassium ion channels open. This leads to a process called repolarization. This occurs as potassium ions leave the neuron, as the voltage-gated potassium ion channels have opened. Repolarization is basically when the potential of the neuron is returning back to a negative state as the potassium ions are leaving. When most of the potassium ions have left the neuron, this causes hyperpolarization. Hyperpolarization is when temporarily the potential or the charge of the neuron drops below that of the resting potential. This is only something that is temporary. When hyperpolarization occurs, the voltage-gated potassium ion channels close. And then a sodium-potassium pump, so the exchange of sodium ions and potassium ions, returns a neuron back to its resting potential. So an action potential is kind of like a cycle. This is the graph that we sometimes use to display what happens in an action potential. So here we have a flat line at the start here at minus 17 millivolts. This represents the resting potential, which is maintained for a short amount of time. Then an impulse comes from the receptors so that the sodium ion channels open. So there is an influx of sodium ions into the neuron, which causes depolarization. So as you can see, the membrane potential or the charge is increasing slightly. And then the threshold of excitation or the threshold potential is reached. So in this case, it's minus 55 millivolts, or you can just write minus 50 millivolts. And this causes voltage-gated sodium ion channels to be activated, leading to an even higher influx of sodium ions into the neuron. So here we have a massive and rapid increase in membrane potential, up to around plus 40 or plus 30 millivolts. which will be the peak action potential, as this is the peak of the graph. After that, the sodium ion channels close and the potassium ion channels open. This causes repolarization, so the inside of the neuron is becoming more negative, so the membrane potential decreases rapidly. Then hyperpolarization occurs, which is when, as you can see, the membrane potential reaches that below the resting potential, as you can see by the graph here. Then the sodium-potassium pump makes sure that the membrane potential reaches resting potential again. So you need to be familiar with the shape of this graph and explaining each of the points. So action potentials are an all or nothing response. So what is an all or nothing response? Basically, an all or nothing response is when once the threshold potential is reached, so around about -50 millivolts, each action potential depolarizes the axon to the same voltage by voltage gated sodium ion channels. So the same voltage is reached each time, which is crucially important in maintaining the rate of impulses. So now we are going to talk about the refractory period. So what is the refractory period? The refractory period is a period in an action potential when the axon can't be depolarized to initiate another action potential. So the axon isn't depolarized, so depolarization does not happen in this stage. This is important because it limits the frequency of action potentials, as we do not want too many action potentials to be fired, as this will be detrimental. Also, it ensures that action potentials only travel in one direction and that they are discrete. Obviously, if action potentials travelled in different directions, this will be a disaster. So now you need to know about the transmission of action potentials in myelinated and non-myelinated neurons. So non-myelinated neurons are basically neurons without a myelin sheath. So in non-myelinated neurons, transmission of action potentials kind of acts like a Mexican wave. I'm pretty sure you're all familiar with what a Mexican wave looks like. This is because when depolarization occurs, voltage-gated sodium ion channels open further down the axon. So, for example, let's just imagine this is a non-myelinated neuron for now. If we have depolarization occurring here, then this could mean that sodium ion channels open further down the axon, so here or here, for example. So by the time the depolarization has spread further down the axon, part of the neuron has already been repolarized. So once the areas further down have been depolarized, for example, then this part of the neuron might have already been repolarized, so returned back to its resting potential. But it's different in myelinated neurons, as action potentials can only occur at the nodes of rambia, which are the gaps between the Schwann cells. This means that action potentials jump from node to node. So it travels from here to here to here to here. This is a much quicker process and is called saltatory conduction. This term here is key, saltatory conduction. So now we're going to focus on the factors affecting the speed of conductance, or the speed of impulses. The first one is myelination. So myelination increases speed, as I've said just earlier now, as saltatory conduction occurs. Another factor is axon diameter. So the wider the axon, the faster the impulse. And finally, temperature. So the higher the temperature, the higher the speed. However, once the temperature has gone past its optimum, obviously enzymes start denaturing. So it's kind of like the same effect as temperature on enzyme activity, which I mentioned in my proteins and enzymes video. If you haven't watched that video, go watch that now in the corner. Right, so that is it for the content, and now I'm going to get on to some exam style questions. So if I just get my highlighter tool out now. So the body loses heat quickly in cold water. A researcher investigated the effect of length of bath time in a bath of ice-cold water on the reaction times of 20 healthy people aged between 21 and 23 years of age. She measured each person's reaction time after being left in ice-cold water for 15, 30 or 45 seconds. She also recorded each person's time before being placed in the ice-cold water, so 0 seconds. So the table shows her results. So here we have in this column the length of the bath time in ice-cold water in seconds, the mean reaction time in seconds, and the standard error. So let's look at the first question. So the first question says, one reason that reaction time is slower when body temperature falls is because nerve impulse conduction is slower. Explain how a lower temperature leads to a slower nerve impulse conduction. Now, as this is an explain question, you need to justify what you're writing. So you need to write why something occurs, not just what occurs. So this is what I've written. I've written that the diffusion of sodium ions and potassium ions is slower. Note that I have not written active transport of sodium and potassium ions, as the question isn't referring to the sodium-potassium pump. So I've referred to sodium and potassium ions diffusion being slower in my answer, as this would slow nervous impulse, as nerve impulses rely on the diffusion of sodium and potassium ions. So let's look at the mark scheme. So the first marking point says slower diffusion, which we wrote, so we would get that mark. Note that the term diffusion is underlined here, so you must put this to get this mark. Also, it says accepted description of diffusion, e.g. movement down concentration gradient, but concept of slower is required. So you need to refer to the term slower or something similar to get the mark. And you can write a description of diffusion to get a mark as well. So the second marking point says of ions, or sodium or potassium. We wrote sodium and potassium, so we can also get that mark. So we would get all 2 marks for this question. Also here it says reference to ions is required. So reject other named ions, for example calcium ions, as these are not involved in nervous impulses. They are involved in synapses. Also it says ignore references to synaptic transmission or rates of respiration, as the question is investigating the rate of nervous impulses. So let's move on to the next question. Other than temperature, give 2 factors that affect the speed of impulse conduction. So it's asking you for 2 factors, but not including temperature. So this is a pretty easy question, as it is just asking you for fact recall. So as I said earlier, myelination, axon diameter, and temperature affect the speed of nerve impulse conduction. But in the question it says other than temperature, so we can put just myelination and axon diameter. So let's look at the mark scheme. So the first marking point that you could write is myelination, or you can write saltatory conduction. Also, it accepts references to dependence on nodes of Ranvier, so you can put that as well to get the mark. But we put myelination, so we would get the mark. Also, it says axon diameter, which we wrote, so we would get all 2 marks for this question. So let's move on to the next question. The next question says, suggest, the key term here is suggest, the conditions that the researcher used when obtaining her data for 0 seconds. So 0 seconds is obviously when the people in the experiment have not been in the ice-cold water yet. So this kind of acts like a control. So this is what I've suggested. Room temperature, as they are not in the ice-cold water, so obviously the temperature must be normal, but kept everything else the same, as it is clear that this is a control. So let's look at the mark scheme. So the mark scheme says keep everything the same but not in the bath, or you put at room temperature. We put at room temperature, so we would get the mark for this question. Or you can write same clothing as for immersion, or sitting in an empty bath, or sitting in water at room temperature. So you can put any one of those to get the mark. Also, it accepts normal or comfortable as equivalent to room temperature, but it ignores references to body temperature. So if you refer to body temperature, you do not get the mark. So let's move on to the next question. So the next question asks us, explain, so you need to justify why you're writing something, not just what's happening. How the researcher could use her raw data, the key term here is raw data, to find the mode and the range. So hopefully from earlier in your education in maths, you should know the definitions of the mode and the range. So the mode is the most commonly occurring result. So for the mode I've written, find the most common result. And the range, you should already know, is the difference between the largest and the smallest value. However, the question is asking you how the researcher could use her raw data, so we do not need to refer to the difference. So I've put for the range that the highest and the lowest times. So let's take a look at the mark scheme. So the mark scheme says, for the mode, find the most common result, which we wrote, so we would get the mark, or the most common time, or the result, or the time that occurs the most. So you can refer to any one of these to get the mark. And for the range, it says the highest and lowest result, or the highest and lowest time. We wrote the highest and lowest times, so we would get the mark. Also, it does accept the difference between the highest and lowest results or times, so you can refer to the difference to get the mark. However, the examiner prefers if you just write the highest and lowest results or the highest and lowest times, as the question is asking you about raw data. Right, that is all I want to say for this video. Thank you very much for watching. If you have any questions at all, big or small, please leave them in the comment section. I'll be more than happy to answer them, and I'll see you in my next video.